So I had the plenary lecture today, and I talked about a few different topics. So the first one was to give a little bit of background on single-cell genomics and translational applications, especially as they pertain to breast cancer tissues. I talked a bit about some of the new single-cell sequencing methods we've developed that have spatial resolution, so that you can pick out individual cells in a breast tissue section and look at pre-malignancies to understand if they are still in the ducts or if they've already started to invade the surrounding tissues. And so I talked about our data on that project, which reported this multiclonal invasion model of invasion, which is a new way, I think, to think about invasion from an evolutionary perspective, but also has important translational implications. The second thing I talked about was our studies on chemoresistance in triple-negative breast cancer, and there we've used both single-cell DNA sequencing methods as well as single-cell RNA sequencing methods to track both genomic evolution and phenotypic evolution in response to neoadjuvant chemotherapy. And what we found from that study, which looked at around 20 patients, a subset of those 8 patients were looked at with single-cell methods in a lot of detail, was that genetic mutations were pre-existing in the population of tumor cells, and when you treat the tumor with therapy, you're eliminating most of the tumor cells, but you select for small populations that then expand afterwards. And that's true for genetic lesions such as copy number alterations. But in terms of transcriptional reprogramming, it looks like the therapy itself is inducing a reprogramming of the transcriptional profiles that cause the cells to be chemoresistant. And we identified several signatures and pathways we think are interesting and could potentially be targeted to overcome chemoresistance, which is a problem for triple-negative patients because about 50% of those patients develop resistance within 1 to 2 years. And the final project was a new project that we've been working on called the Breast Cell Atlas. And this has nothing to do with cancer directly, but it's you can think of it as a human genome map of all the different cell types in the normal breast, including the epithelial components, so luminal and basal, but also the normal immune cells, as well as things like fibroblasts and endothelial cells that are part of the vasculature, and trying to create a reference of what the normal transcriptional programs and epigenetic programs of those cell types are, so that when we start to study cancer, and we want to understand not just the tumor cells, but the microenvironment, we know what a normal reference should look like. And so we can say that in these early lesions, maybe in DCIS, early breast cancer, we can see changes in fibroblasts, but maybe not changes in other cell types. And we can look at those pathways that are being co-opted and changed, and they can have clinical significance. So it's a kind of a team science project to really have this nice reference of the cell types in the human breast, and it's a bit more related to kind of normal biology as well as normal variation across different women, because we think there's a lot of biological variables that are important, such as menopause, pregnancy, breast size, breast density, those different factors that can affect the cell types. So we developed a method called topographic single-cell sequencing, which allows us to, the problem is that most single-cell methods developed so far, genomic methods, they require that you dissociate a tissue into a cell suspension, and you lose all the spatial information. You don't know where every individual cell came from in the initial tissue. So this is a method that's compatible with H&E sections from pathology. So you can have important pathological features, morphologies, ducts, lobules, different structures, and then you can pick out individual cells from those structures and study their genomic profiles. And this was, we looked at synchronous DCIS, IDC, so that means they both have areas that are in situ and still within the basement membrane of the ducts, and it has areas adjacent that are invasive. And we could study this transition as the tumor cells go from these premalignant lesions in the ducts into the invasive areas and start to expand to form the tumor mass. And so by tracking copy number changes but also mutations, we found that most of the action happens in the ducts prior to invasion. And so this led to the idea that early pre-programming of the genome in terms of copy number aberrations and mutations are likely to cause those tumor cells to either remain indolent for the lifetime of the patient or cause those tumor cells to become invasive later on as they continue to progress and expand. One analogy that people like to use that I did not come up with myself is a fruit smoothie. So most TCGA studies, tumor studies that you see are going to be, you take a tumor, you grind it up, you put it in a blender, and then you get this mixture of lots of different DNA and RNA, and you sequence everything. And you try to figure out what's in that mixture of DNA and RNA. Of course, a lot of times it's half of it's stroma, half of it's tumor. There's a lot of diversity within the tumor cells. There's a lot of diversity within the stroma. There might be immune cells. There might be blood coming in. Single cell sequencing is more like a fruit cup, where you have you can tell what the berries are, or the strawberries, or the different components, and you can determine which mutations or transcriptional programs are associated with all those different fruits that are essentially in the mixture. So it gives a very granular resolution of a tumor, and it allows you to kind of remove effects of cell types that you might not be interested in. So if you want to look at how is a fibroblast in a tumor different than a normal fibroblast, you can do a cell type specific expression, differential expression analysis, which just hasn't been possible before. With DNA, you can look at individual cells and you can completely resolve the heterogeneity. And even though you can get a list of mutations from a tumor, this will tell you which combinations of those mutations are present in any given cell or subpopulation within the tumor. So, and then the other thing is just to be able to really study rare subpopulations that haven't been amenable to genomic methods before. Not just circulating tumor cells, but also things like cancer stem cells or other rare subpopulations. So this is consistent with what we call punctuated evolution, which we've been studying for some time in copy number, where we think these early initial bursts in genome instability can give rise to lots of different clones, but then is turned off later on, and then those clones that are generated and are viable and have a fitness advantage, they will continue to expand and will eventually escape the ducts. So it's this data in pre-malignancies, DCIS, where there really isn't a lot of genomic data so far, is consistent with kind of the evolutionary models we've inferred from the more invasive cancers, which I think is good news. But again, it was this study was about 10 patients, and it did focus on synchronous DCIS and IDC, and I think a better future direction and an even better study would be to have pure DCIS samples and then recurrent samples 5 to 10 years later, where the patient comes back and has IDC, and then you can kind of look at patients that progress to IDC and patients that remain pure DCIS for their whole lifetimes, which I think is better than the synchronous samples. And the problem, though, is that all this material is FFPE, and that's been very difficult for single-cell genomics. So we've been doing a lot of effort and work into developing single-cell DNA sequencing methods that are compatible with FFPE tissues, so that we can do studies like this. I think the question is, how far along are these technologies? Are they translational at this point in time, or are they still kind of only in the expert labs? And so for single-cell RNA sequencing, it's been heavily commercialized because it's being used not just in cancer research, but in neurobiology, in development, immunology, pretty much every field. So a number of companies have 10x Genomics, Biorad, a number of companies develop methods to do very high throughput single-cell RNA sequencing that are based on papers that came out in 2015 that use droplet-like technologies to encapsulate beads and cells and then be able to profile 10,000 cells in one experiment. So that's brought the costs down a lot and has also kind of democratized the methods, and now they're kind of widely available and it's becoming a standard molecular biology tool. Single-cell DNA sequencing has been a little bit harder to develop the methods. It requires multiple steps in chemistry, and unlike RNA, where you have lots of molecules that you can amplify, DNA, you're usually dealing with a diploid genome, which is 2 molecules, and you don't have the poly-A tail, which is used for a lot of the chemistries. So, but single-cell DNA sequencing has come a long way. We reported the first method in 2011, and it has been commercialized also very recently, just in the last few months or last year. And so it's very interesting, I think, to see as these technologies go from expert labs like our lab into kind of the clinic and into more research labs around the world, what types of applications they'll be used for. So I think it's a pretty exciting time. We're kind of going past the initial technology development phases, which have really taken almost 10 years or so, into much more stable technologies, and now we can think about applying them on large numbers of hundreds of patients, and also in a kind of a clinical framework where you have a system that can easily be run by a technician or someone else. And so I think there are some important translational applications, both in looking at circulating tumor cells to do longitudinal monitoring of therapeutic response, also to look at scarce clinical samples, such as fine needle aspirates in the breast or core biopsy samples, to do immunoprofiling, microenvironment profiling, and also to kind of guide targeted therapies, because you can get very detailed maps of the clonal diversity in the tumor, but also kind of the lineage of the tumor cells, and you can make decisions about, do you want to target a driver mutation that was acquired early and was inherited by all the tumor cells so that you can target everything, or is there a subpopulation that you think is going to be more metastatic, and should you only target that population? So I think there are a lot of exciting clinical applications, and us and other groups are starting to work on projects that use these technologies. Circulating tumor cells is probably the one that's going to be the most translational first, just because there are good techniques to do enumeration, and there are also pretty good techniques for picking out the CTC, so now we have the techniques to actually do the single cell genome or transcriptome profiling. So. No, I don't think so. I think it's an exciting time, and I think these technologies have matured now to the point where we can start looking at large cohorts of patients, using them to analyze samples from clinical trials, and really gives us a lot of information. So